Hello, everybody. Good morning. We're going to leave a few minutes in order to let remaining attendants join us. So thank you for waiting, and came back in 2-3 minutes. Okay, we're going to start. So good morning, everybody, and welcome to our first Condalab Talk Innovation Series 2022. From Condalab's team, we want to say thank you for your attendance, and we hope you find today's Condalab Talk interesting. First, I'm going to introduce myself. My name is Jaime Martinez, a microbiological product manager at Condalab. I am a biologist, and I joined to Condalab in 2016, 6 years ago. I'm telling that because from those days to nowadays, I have seen this company improve and evolve a lot until today, when we are here to present our new line Condagene, another new milestone that shows the commitment to innovation that Condalab has always showed. Condagene is a product line of qPCR detection kits of Condalab, focusing on offering a competitive and rapid solution to microbiological laboratories in food safety and environmental monitoring matters. Without further ado, let's see what we have prepared for today's talk. The estimated duration is 30-35 minutes. I will try to be as brief as possible to leave time for questions at the end of the speech. You can send your questions through the display that you can see in your right side of your screens in the question seconds, not chat, please, because maybe all questions will not be answered just because time matters. So we will answer all of them by email. So don't worry, you put your question in this section and we will answer all of them. Okay, so I'm going to put off my cam and let's see with presentation. Well, here we have the index of what we will see today. I know that many of you will be familiar with PCR, but since the public and the audience is quite heterogeneous, it have different background, allow me to dedicate a few first points to define the principles of these techniques and its advantage that its use present for a microbiological control laboratory. Then, after discussing about some limitations, we will see how Condagene is the ideal tool to provide solution to those points, as well as how our kits present all remaining advantages that this technique has. Bigger advantage than its limitation, everything must be said. Finally, I will show you our catalog, which Condagene have been launched. We do not want you to think that it's the end. We did the impression that there are a few products because it's a new line that has just been born, and we are working a lot to launch new products every year. Without going any further, hopefully, we believe that turn to the summer, we will present new developments. Although all of us already know what these development issues are like, and sometimes we don't know what we'll find. Well, so let's go starting by what the PCR technique consists of. PCR is a very powerful and transversal diagnostic tool. These technical principles are based on DNA replication capacity. Where using different reagents, we will be able to obtain a huge number of copies of the same DNA fragment. These reagent or component are first the target DNA, which will be amplified. Second, we will need some primers, which are small, known, and complementary DNA signal to the ends of the DNA region that we're going to amplify. With these primers, we are delimiting the DNA area which we want to detect. Finally, we will also need nucleotides, units constituting DNA, and one enzyme on charge to add these nucleotides and amplify the target DNA fragment. PCR consists in several consecutive cycles where the same steps at different temperatures are repeated one and another thanks to the DNA behavior depending on this specific temperature. At first, the denaturation of DNA is carried out, that is, the two strands of the double helix are separated. This allows, in a second stage with different temperature, the hybridization of the primer to their specific region, the complementary, of course. After that, once the fragment to be amplified has been delimited by the primers, the third step starts, amplification. That happens thanks to the action of the polymerase enzyme that will be incorporating nucleotides and making 2 copies of each fragment. So with this cycle is repeated n times, so copies generation occur exponentially, allowing reveal their presence. Well, we have just seen will be the basic PCR technique, but there is another technique called qPCR or real-time PCR, which allows through fluorescent detection to monitor amplification in real time and even quantify the amount of DNA. This is possible thanks to the use of thermocyclers and fluorescent DNA dyes. There are 2 types of dyes for qPCR, specific ones of double-stranded DNA or specific for target region. The latter, known as proofs, are the ones we use in Condagene kits. They are much more specific and work thanks to the presence of a quencher and a reporter. These molecules are attached to the proof, and the quencher inhibits the fluorescent emission of the report. Once the polymerase amplifies the DNA, it is removed, and the quencher is removing, and then the reporter emits the fluorescent that is kept by the equipment. In this graph, you can see how the results of these solvents in a qPCR thermocycle. We can see the fluorescent emission on this axis, the vertical one, and the number of cycles in this one. Positive result will be when this kind of curve appears on the graph, where the increasing in fluorescence is so in what number of cycles is happening. I mean, and using these standard curves, the fluorescence could be quantified, making qPCR not only a qualitative technique like conventional traditional PCR, but we will also will be a quantitative one. Okay, so once I have tried to explain what is the qPCR technique and what are its principle, let's see what advantage it would bring to our microbiological analysis laboratory. At first, we're going to see what the workflow is by using traditional methods with culture media. We have a first sample dilution and treatment step, which can take about 24-48 hours, depending on the procedure. Next day, there may be other secondary selective enrichment step, but to the necessity of microflora sample inhibition. The work day after, we will do the insulation in a selective agar. If we have growth through the isolation of suspect colonies, we will purify these colonies in a new non-selective agar, and after, again, it's incubation. We will do the confirmation test to see if the suspected microorganisms are target or not. In case we test by PCR, we will have next protocol. The first step starts with the same one as culture media isolation with a sample treatment according to the specific standard. After this dilution, we must extract the DNA present in the dilution broth, usually buffer and pet of water, for example. Sometimes it's also interesting to do a short selective enrichment around 4 or 5 hours on the same day that we will do the extraction to enhance the presence of our target microorganisms. DNA extraction is a key and necessary stage, since we need to release the genetic material which are inside the cells. To do that, we need specific reagent which will allow the cells to release the DNA to the media. This stage usually takes between 15-40 minutes, depending on the protocol. Once the DNA is extracted, we will prepare the PCR plates, mixing this DNA extracted from the sample with all the necessary reagents for qPCR. Then the test plate will be introduced in the thermocycle, and after a few hours, we will have present absent results. So what will be the first and obvious advantage? Clearly, time to results. Since depending on the protocol, we could save days of work. But it's not the only advantage that PCR methods can bring to your lab. In addition to the decreasing of time, which I said in some cases can reduce up to 10 days, as for example, in Legionella testing, qPCR testing allows to the industry the positive release of batches. Thanks to the use of a specific sequences improved, absence result can be releases without confirmation. This gives to the industry and the labs a quick response covering the most probable results. Do the control and prevention plans together with the quality guidelines ensure many times the absence of these pathogens. On the other hand, this technique has a high sensitivity and its detection limits very low. The theory says that with a single copy, you will be able to amplify it. Obviously, in practice, it's necessary to inoculate something more from the extracted to ensure that the copy is in the analyzed sample. Another advantage would be the specificity based on exclusively DNA, so the associated microflora will not interfere. The use of commercial qPCR kits presents simple and straightforward protocol with a minimal handle would reduce the rate of human error. Finally, this technique allows automated points during the procedure, providing great value to those laboratories which have a huge sample volume or which want to minimize human manipulation to avoid as much as possible the variability source. But this technique presents some limitations, which in certain way are limiting its implementation in some laboratories. Differentiation between viable and non-viable cells is one of the main limitations of qPCR. Unlike culture media, in which only viable cells grow, PCR are detecting DNA fragments, making no difference between if the bacteria are alive or dead. This makes it possible to have false positives in our tests. Another limitation, in this case logistic one, is the need for a cold transport for this kind of product, because it's necessary for the liquid solution be shaped in dry ice, making transport more expensive and presenting some risk of breaking the supply chain. Another point to consider is the cross-contamination that usually appears in witches, and in which is one of the most important points when it comes to optimizing and giving robustness to our results. It is unfortunately very common in molecular biology laboratories, which require very rigorous work protocol. And these applicants can run later and give us false positives, in case we can't not remove it. Finally, although quantification is possible thanks to qPCR, it must supported by a validation assay, because there is no one-to-one relationship between genomic units, PCR results, and CFUs, which is the measure in which results will be expressive in quantitative microbiological control. So now we're going to present Condagene, which try to solve many of these limitation points that I just seen, and combining with the advantage of qPCR. So first, it should be noted that Condagene present control points according to ISO 22174 standard. This is a specific standard for the use of PCR in the detection of pathogens, determining the use of negative and positive control. In addition, Condagene presents on internal control already including the kit that offers protection against false negative. Because if it is not amplified, something will be inhibiting PCR and we will take some correct measure. Also, our kits have two protection tools against false positive. The first one is UNG control added to our kits. This eliminates or remains present in over amplifications runs by degrading uracil molecules which are nucleated usage in PCR products, and it is not naturally present in some. In this way, degrading, removing uracil molecules, we will prevent this fragment from being amplified. Condagene kits are also compatible with viability PCR, and in a specific reagent, it binds to free DNA and block its amplification. This step must be done prior to extraction. We don't have this reagent in our catalog, but it could be used with any other reagent on the market. Anyway, as I mentioned before, Condagene has just been born, and there are many paths that we are traveling, and that soon we hope to make them available to you. Condagene, according to the ISO 22174 standard, thanks to the used specific proofs included into the kit, would not require confirmation. We already know that the rules and standards sometimes are very ambiguous, so that is why we will recommend confirm the positive case in plate. Well, So more advantage about Condagene. Our kits are sample and have direct protocols which ensure minimal handling. On the other hand, Condagene has a very interesting point, which is that these kits are designed with high stable liquid compounds, allowing them to be shipped at room temperature. We have done some studies which demonstrate the performance of our products after 14 days at 37 degrees. So this will allow saving costs in transportation and avoid risks such as the breakage of the cold chain that we mentioned in previous slide. These products are also open kits compatible with many thermocyclers on the market. Of course, we have not tried absolutely all of them because, as you already know, there are a huge offer on the market, but we have tried Condagene, the most important one. We are increasing our thermocycler database, but if you have the right reaction tube and your software allows to set the necessary protocol, you should have no problem using our kit with your equipment. Our interesting point is that Condagene kits present a positive reading at a homogeneous number of cycles, so we can use different reagents or different detection kits on the sample on the same plate, of course in different wells, allowing us to carry out kind of multiplex, identifying different parameters in the same plate, okay, not in the tube, that will be the correct multiplex term, but for many labs will be so useful. Another advantage is that our products have been developed according to standardized methods, being ISO FDA, which voids the necessity to accredit them and will require just a verification process in the lab. If the entity that is going to implement Condagene is accredited by the ISO 17025. All our kits have, finally, all our kits have a price per sample, including extraction, which is very competitive compared to other kits on the market. So, and you can see the advantage of Condagene's implementation in your lab are many, and our versatility allow us to adapt to the needs that each customer has. Now, let's go with our Condagene catalog. Well, today I'm going to launch 4 different products. We have available 2 DNA extraction kits and 2 pathogen detection kits, Salmonella and Legionella. We're going to share to see their characteristics and protocols, but before, I want also to share with you a new launch. It's also that is our thermocycler CDL96. This thermocycler is with a Condalab brand. It's an open system and is so versatile. I mean, all Condagene kits are validated and proved and optimized with our thermocycler. Okay, and say that Condagene is open system and it is, but we are working also all the development and everything with this thermocycler. Has a high sensitivity, high accuracy optic system, has 4 channels. There is also another option for 6 channels thermocycler, has an intuitive and user-friendly software, touch screen, big touch screen, high temperature control system, many different data analysis functions. So if you, okay, I'm not the product specialist of this equipment, but anyway, if you have interest and you know more about it, please let us know and we will keep in contact in order to discuss about this topic. So first, I'm going to show you the extraction kits. Our extraction kits are based on lysis buffers. The goal, as we saw in our previous slide, is to break the cells in the sample to obtain free DNA that can later be amplified in PCR reaction. These kits are specially designed and optimized for this use with Condagene detection kits, obviously. If we do that, we will find a minimal food magic influence and getting very low inhibition rates. Also, our kits are easy and quick to use. These are available to different kits. The first one, the CATS 6500, has 2 different reagents, different bottles. So this kit is very effective for those matrices which have a high soil load and also can be used in both positive and negative gram bacteria. The second one, CAT 16501 Condagene Quick, which allows a single-step extraction with only one reagent, but it is specific for gram-negative bacteria. In addition, its effectiveness makes the signal reading much cleaner and clearer. What will be the protocol? Okay, for the extraction kit Condagene, we can say our universal extraction kit. Here we will start with 1 milliliter of the previously enriched sample. Then we will centrifugate it at 10,000 RPM for 10 minutes. Once this has finished, we will remove the supernatant and we will resuspend the pallet with 0.2 milliliter of reagent number 1. We'll take this suspension to the thermoblock and program it at 1890 degrees at 1000 RPM for 5 minutes. Once this is done, we add 0.6 milliliter of reagent number 2 and cool the thermoblock at 20-30 degrees for 2 minutes and 1000 RPM. Finally, we will take it to a vortex for 50 seconds to homogenize the reagents and centrifugate it one last time at 10,000 RPM for 2 minutes. After this, we will have the supernatant, which will be our sample ready to load the PCR. Well, if that seems simple to you, let's now see what the protocol will be like with Condagene Quick Extraction Kit. In this case, we start with a smaller amount of sample between 0.2 0.5 milliliter. We'll centrifugate it for 10 minutes at 10,000 RPM. Then we will remove the supernatant and resuspend the pellet with 0.2 milliliter of the reagent, the only one in this case. Once resuspended, we took it to the vortex for 15 seconds with subsequent centrifugation for 2 minutes at 10,000 RPM. And with that, our sample is ready for PCR. These products and all Condagene's products, as we will see later, have all the bibliographic material and validation reports at your disposal. These kits, for example, have been tested with both heat-treated and non-heat-treated complex matrix, and also in raw meat or processed food, for example. Now, Condagene Salmonella SPP, indicated for the analysis of the presence and absence of Salmonella SPP in food samples. Let's see first what is the current method by culture media to put in perspective. Well, ISO 6579 methods indicates a first enrichment step in Waffen-Pepton water. To do this, we would take 25 grams of the sample to 225 milliliter of broth, incubated that for 24 hours at 34-38 degrees. The next day, we will carry out a second selective enrichment for 24 hours in parallel in Rapaport Bacillus broths and also in MCAT-TN. These broths will be incubated at different temperatures. 41.5 the Rapaport and 37 the MCAT-TN. After 24 hours, we split them on XLD selective agar and on another secondary selective agar with other selective agents different than XLD for another 24 hours. Already on the fourth day, the suspect colony should be purified in non-selective agar to later carry out auto confirmation tests. In total, 5 to 7 days, 6 different culture media at least, and all confirmation tests serological reagents. Thanks to Condagene, you can reduce. Thanks to Condagene, you will reduce to do 2 days and 3 different reagents. Of course, you use our quick extraction kit. Moreover, to all the advantage seen through today's talk, Condagene's method is based on the technical specification for German government and in accordance with the German standard DA in 10135. This method also was evaluated in an interlaboratory trial when 13 participants were supporting the method. This protocol starts with an enrichment according to ISO 6579 in bath-and-peptone water. Once incubated, we will inoculate 0.1 milliliter of the bath-and-peptone water in 10 milliliter of Rapaport bacillari ISO formulation and take it to the incubator for 5 hours. If we do that in the morning, we can already organize ourselves to prepare PCR in the same day. Once the sample is enriched, we will take the extraction according to the corresponding protocol, which will take us between 15 or 30 minutes. When the extraction is done, the PCR will not take more than 3 hours to have the results. On this slide, we have more information about the kits. The content consisting of 4 different kinds of tube. Each one have one with with a different color that allows its identification and facilitates the follow up the protocol. Green cap contains master mix, which comes in liquid format. In this tube, we include controls, internal ones, and UNG, polymerized, the nucleotides, and other components. We also find a tube with a blue cap, which is including the proof and primers in lyophilized format. The positive control will be added to the yellow cap, and the PCR grade water come in 2 tubes with a white cap. So how we will proceed to prepare the reagents? Well, the first step is to reconstitute the positive control. To do this, we add 100 microliters of PCR grade water, white cap, to the control tube, yellow cap. It is necessary to shake cap 5 times with the pipe. Doing that, our positive control will be ready. On the other hand, we need to reconstitute the primers and probes. And we're going to add all tube content of master mix, green cap, to the tube containing the proof and primers, blue cap. As in the previous case, we will shake 5 times with the pipette, then our master mix proof primers will be ready for PCR. So how do we load the PCR plate? Well, in one well, we must include the positive control, in another one the negative control, and in another the sample. We can run multiple samples at once if we include and identify positive control. The negative one will be always the same. In each reaction tube, we load 4 microliters from the blue tube, which contains proofs, master mix, and primers, 11 microliters of PCR grade water, white cap, and 5 microliters of sample. If we are doing the sample is the extraction, the supernatant of the extraction tube. So we will load 5 microliters of sample, or 5 microliters if we are doing the positive control, and from the yellow cap, of course, and 5 microliters of negative control, that is the white cap. Once all the reagents have been loaded, we can start up the thermocycler with a specific setup for each kit, which is indicated in the instruction for use. The entire content of the kit, both extraction and detection, we can do 100 reaction or 100 extraction. Now, I'm going to introduce our last product, Condagene Legionella SPP. At the same way than the previous case, we will first review how ISO method is. A FISO standard method for Salmonella could be a bit long. Let's not talk about the one of our Legionella. As many of you already know, the method is very long and tedious, needing around 15-20 days to obtain results. We did a specific Legionella Condalab tools where we deal with all the details of the method, complex as I say. Here, I will try to, the previous Condalab tool from Legionella is more extensive, we try to deep in all the different aspects for the method. But here I will try to capture the main bottlenecks. First, we should do we will with the procedure. First, we should do a pre-treatment sample depending on its origin and type. These samples will be divided into sub-samples that will receive these different pre-treatments and treatments. Then we will filter the sample through, and in some cases might require prior elution before spreading it in a battery of selective media with different mixture of antibiotics. We are talking about 4 different media. Growth will be monitoring over 7 to 10 days. After incubation, subspecies colonies will be spread in a minimal agar that also requires a long incubation, often up to 5 days of incubation. And then we will do the confirmation also with serological proof, and then we will get the results between 15-20 days. Complex and long, right? Well, thanks to the Condagene method, based on the ISO standard 12896 for the detection of Legionella by PCR, with this method, you will be able to drastically reduce the handling of reagents and improve the time to result in just 2-3 days. For the protocol, very similar to Salmonella SPP, we will follow the indication of ISO 11731 for the treatment of the sample. Once treated, we carry out the filtration and carry out the extraction on the diluate. Then we'll load the plate with the necessary reagents and take it to the thermocycler, and in approximately 3 hours, the result will be ready. So, Salmonella, Condagene Legionella SPP, packaging and reagent are the same than Condagene Salmonella. There is, there is the same core classification system allowing us an easy preparation of reagent. Obviously, in this case, positive control profs and primer will be different from Salmonella, but we find the master mix in the green cup, the profs and primer in the blue cup, the yellow cup for positive control, and the white cup with PCR grade water. Remember that master mix comes in liquid format, and the proof, primers, and positive control are in lyophilized. Well, reconstitution is the same, only with one difference. In this case, we recommend leave the reagent around 15 seconds at room temperature, and then centrifuging to 15 seconds at 5,000 RPM to ensure the homogenization. On the other hand, this kit is indicated to quantify the results, making a standard cube using the positive control for it. How to do that is reflected in the technical documentation. Both for this case and for Salmonella kits, we recommend aliquoting into tubes according to the needs and dynamic of the laboratory, because all these kits have to be reconstituted at the same time. And since once reconstituted, we will have to keep them to minus 20 degrees to preserve. Of course, the reagent have a freeze-thaw study, and the number of cycles is limited and indicated also once again in technical specification. Finally, the loading of the tube is also the same. 4 microliters from the blue cap, which contain probe master American primers, 11 microliters for PCR grade water, 5 microliters from the sample, from the positive control or from a negative control. Also, for the product, the quantity included in Condagene Legionella it is indicated for 100 reagents. So all this that I have told you is supported by related technical documentation, as I mentioned along all the presentation, which is available in Condalab. Here we will find technical datasets, safety datasets, certificate of analysis for each product. In addition to this elemental and basic documentation, there are available the instructions for use. It is very detailed document, including the protocol with all the information related to standard, required equipment, operation procedures, thermocycler setup, or including uneven interpretation of the results. Likewise, we have verification report for each kit, indicating results in inclusivity test, tested matrix, and studies conclusion. Finally, you will also have available in different studies, different solvent stability studies, both for transport as well as for the freeze-thaw cycle and for the shelf life, which is currently 8 months. But which is I'm talking about the shelf life that is currently 8 months, but we're waiting for result for 1 year that we are convinced, we are sure that we will arrive. Okay, so that is the end of my presentation. What time is it? It's 10:42. We are going to have 5 minutes of question. So we are going to try to learn them. Okay. Okay, let's see if there is any question. Yeah, as I said, we are running a little tight sometimes, so we have some of them, but no worry, because remaining question will be answered by email. So I'm going to share the screen again. Okay, so here we have seen that your kids have control for false positives, but how do I detect an inhibition in the PCR? Okay, so false negatives are monitored thanks to the presence of an internal control, which is an amplification peak before the reading. In other words, if we are seeing no amplification in the internal control, something is falling with the PCR. Perhaps many times an excessive amount of DNA. In this case, it would be enough to delete sample 1 to and decimal dilution to obtain a signal. If we have amplification in internal control but not in the sample, we can ensure that there is absence of that microorganism. So no, if internal control don't be amplified, something is wrong with the PCR. So that's why we try to with this tool, we fight against the false positive. Sorry, the false negative. Okay, we are going to read one more. Can I keep the reagents in the fridge? Of course, as I said, reagents are stable at room temperature, so storing a refrigerator for 1 week would be possible. Okay. Despite this, we do not have studies on this regard, but storage conditions and good laboratory practice lead to keeping this product at -20. Okay, one more question. Can I use part of the reagents and then ship them? Yes, of course, they can be used. They can be used later, but their preparation, as I said, must be in one time. Knowing that, the ideal situation, we will prepare everything together and then aliquot into single tubes depending on the sample volume. This should be kept at minus 20 degrees. Okay, another one. What matrices are validated for Legionella? Well, for Legionella, we have validated samples from cooling towers, sanitary and hot water. Sanitary hot water, sorry, and drinking water. And I say that this is not the question, but I take this opportunity. And for Salmonella, should be consulted because there have been quite few metrics that I remember: dairy products, raw material, fresh meat, bakeries, egg products, and so on. In any case, all these are indicated in the documentation, which is available for your inquiry. Okay, and one more, the last one. How many PCRs can I run with the amount of positive control? Okay, like everything, it will depend on the need of each one. If we follow the guidelines to restitute with 100 microliters of PCR, PCR grade water, are indicated this one. So if we use 100 microliters and load 5 microliters in the well, it will be as 20 runs per PCR. Of course, you have also to know that this quantity of 100 microliter are indicated to reach a known dilution to carry out the standard curve for quantification. So if we only need to detect presence or absence, we can reconstitute the control with 250 microliters, which will give us about 15 runs more. Okay, so time is over. We are out of time. This has been all from our side. I hope you have found it interesting, and we are at your complete disposal for any question. Be sure to visit our website to get more information about Condagene. Also, I don't want to miss this opportunity to share with you the enthusiasm that this new line of products represents for Condalab, and in which we continue working to bring new kits to the market that cover all your necessities and providing you with improvements in your day-to-day. So stay tuned because news will come. Thank you so much for your attention. Have a nice day and hope to see you soon in the next Condalab. Bye bye.